In this video, we'll be looking at the immune system part 2, adaptive immunity. There are many different types of immunity, or in other words, resistance. The two major types of immunity are innate immunity, or nonspecific resistance, or adaptive immunity, also known as specific resistance. This is a flowchart showing the many different mechanisms used in innate immunity versus adaptive immunity. And in this lecture video, we're going to be focusing on adaptive immunity, or the specific resistance. This is also known as our third line of defense. There are two major types of adaptive immunity. This includes cellular immunity, where the primary effector cells are T cells. And then we have humoral immunity, where the primary effector cells are B cells, which make proteins that help our body recognize foreign antigens known as antibodies. Before we look at the difference between cellular and humoral immunity, let's review the difference between innate and adaptive immunity. Immunity is also known as resistance, and this is the ability of the body to resist infections by pathogens. We have two major types of immunity. We have innate immunity, also known as nonspecific resistance, and we have adaptive immunity, also known as specific resistance. Innate immunity is called innate because this is the part of our immune system that we are born with. Whereas our adaptive immunity actually changes over time as we are exposed to different pathogens. Innate immunity is a collection of defense mechanisms. For example, this includes our external barriers like our skin or mucous membranes, as well as certain white blood cells, proteins, and physiological processes like inflammation and fever. These mechanisms guard our body from a broad range of pathogens and are not very specific. Our innate immunity and these mechanisms do not develop a memory of a pathogen. Therefore, it does not become more effective at fighting a pathogen even after multiple exposures to that pathogen. Also, the effects of our innate immunity tend to usually be localized. An exception to that would be fever, which is more systemic. Now, when we look at adaptive immunity, or in other words, our specific resistance, this is a collection of lymphocytes, T cells and B cells, as well as antibodies, which are produced by B cells, that become specialized to fight specific pathogens. This is why this is known as our specific resistance. Now, this is also known as our adaptive immune response because it develops a memory of a pathogen and becomes more effective at fighting that pathogen after multiple exposures. So it adapts over time. Since antibodies circulate in our blood plasma and lymphocytes are distributed throughout our lymphatic organs, such as in our lymph nodes as well as our mucous membranes, the effects of our adaptive immunity tend to be more widespread, or in other words, systemic. Now let's focus on the two different types of adaptive immunity. This involves our cellular immunity as well as our humoral immunity. When you think of cellular immunity, you could think of it as cells against cells. This is going to involve our T lymphocytes, also known as our T cells. T cells destroy virally infected cells, foreign cells, and disease cells. So this is an example of cells killing other cells. Our cellular immunity is also a way that our body can attack intracellular pathogens like viruses. Now let's look at humoral immunity. Humoral means bodily fluids, and that has to do with the fact that during a humoral immune response, specialized B cells, called plasma cells, will secrete antibodies into our bodily fluids, and these antibodies will circulate throughout our blood plasma and therefore help our body recognize foreign pathogens. Antibodies are proteins that can bind to proteins on pathogens and help our body identify those. Antibodies bind to these proteins known as antigens on the pathogens, therefore tagging them for destruction. Antibodies can only bind to extracellular antigens. So if those antigens were present within a cell, the antibodies would not be able to bind to them. There are different methods by which our body can acquire adaptive immunity. And when a person acquires adaptive immunity against a pathogen, it means that their immune system can recognize that specific pathogen and destroy it quickly before that person ever gets sick. So let's look at the different ways that our body can acquire adaptive immunity. There are actually 4 ways of acquiring adaptive immunity. We have naturally acquired passive immunity, artificially acquired passive immunity. We have naturally acquired active immunity, or artificially acquired active immunity. So let's first look at what is the difference between passive immunity versus active immunity. And what do we mean when we say that it's acquired naturally versus artificially? Passive immunity requires our body receiving antibodies that can therefore help our immune system recognize a pathogen. Remember, antibodies are proteins that help our immune system recognize pathogens. This is only a temporary form of immunity because once those antibodies break down in our bodies will no longer have immunity against those specific pathogens. Active immunity, on the other hand, requires the body to be exposed to the antigens found on the pathogen. Okay, so it requires our body to be exposed to these foreign proteins known as antigens. This will therefore allow our body to actively create memory B cells and T cells. And these memory B cells and T cells provide a memory of that pathogen in our body and therefore provide long-term immunity because B cells and T cells can exist in our bodies for decades. So let's look at the 2 ways that we can acquire passive immunity or active immunity. Now, we can acquire passive immunity either naturally or artificially. So let's think about it. How could you naturally acquire antibodies? How could your body naturally receive antibodies? Well, this tends to only occur 2 times in your life. Antibodies are naturally acquired through breast milk. So breastfeeding helps to improve the immune system of the baby by actually giving specific antibodies to the baby to protect them against certain pathogens. So this is why whenever a mother is breastfeeding her baby, as long as she's breastfeeding her baby, there's a lower chance of her child getting sick from like a common cold or something like that. In addition to that, the placenta helps to transfer antibodies from the mother's body to the fetus. Okay, so that is another way where antibodies are naturally given to another individual, from one individual to another. Now, how can we artificially acquire antibodies? Well, this can happen through injections. So injections can contain antibodies. Some examples of this include antibody serums. So it's possible that you actually receive a serum that contains a specific antibody that can help your body recognize a certain pathogen. Also, antivenoms. The way that they actually work is giving your body antibodies against a certain toxin. So if you get, for example, bit by a snake, you might receive an antivenom. The antivenom contains antibodies to help your body neutralize those toxins. So these antibodies will bind to the toxins, preventing them from making you sick. Also, another example would be the rabies post-exposure shots. So if you actually get exposed to the rabies virus, you might be given post-exposure shots. And what is actually in those shots are antibodies. Those antibodies help to neutralize the rabies virus, and it actually will bind to the antigens on the rabies virus and prevent that virus from being able to infect your cells. Now let's look at how active immunity can be acquired. So remember, active immunity involves our body being exposed to the antigens or the foreign proteins on a pathogen. This is actually going to trigger your body to produce memory B and T cells. Now, we can naturally be exposed to the antigens found on a pathogen, or we can be artificially exposed to these antigens on a pathogen. So how might you naturally be exposed to a pathogen? Well, that would be from being exposed to a pathogen from another person and getting sick. Okay, so you might actually catch a specific pathogen or virus or bacteria from somebody else. An example of this would be catching a cold, the flu, or COVID from being exposed to somebody else. When you're exposed to those pathogens, your body will naturally respond by creating memory B and T cells to help your immune system be better at recognizing that pathogen the next time you're exposed to it. Now, how may you artificially be exposed to the antigens found on a pathogen? Well, that is through vaccinations. So that's how vaccinations work. Vaccinations are an artificial way of acquiring active immunity. Vaccines expose you to the antigens of a pathogen so your body can learn to recognize that pathogen before you are actually exposed to it. There are a lot of different types of vaccines, but let me just introduce you to some of the different types of vaccines and how they may be introducing the antigens from a pathogen to your body. Okay, so here we have a virus. A virus is simply just a protein shell with some type of genetic material in it. It could be DNA or a small segment of RNA. And then surrounding that, you'll have like a lipid envelope. And then there's usually these proteins sticking out of the surface of it. These are what we call the viral antigens. Those proteins or viral antigens are what your immune system are responding to in order to develop your adaptive immunity. So some vaccines actually contain inactivated or weakened viruses. Therefore, that's exposing your body to all the viral antigens. So one virus could have many different antigens on its surface. By exposing your body to an inactivated or killed virus, your body will then be exposed to the antigens, but you won't get sick because that virus has actually been killed or I shouldn't say killed because they're not actually living, but that virus has been destroyed in some way, probably by like heating it up or something. So anyway, you're exposed to that inactivated or weakened virus. That exposes your body to the viral antigens, and that triggers your immune system to create memory T and memory B cells that are capable of recognizing all those different antigens on that virus. So this would allow your body to create many different memory B cells and T cells, and each one of those different memory cells can recognize different ones of those viral antigens. Another type of vaccine may contain parts of a virus, or in other words, the antigens of the virus. So instead of containing the entire virus, that vaccine only contains maybe one or two of those specific antigens. So those viral antigens are then injected into your body. Your body will recognize those as foreign proteins and therefore create memory T cells and B cells that can recognize those viral antigens, so that if you are exposed to the actual viruses in the future, your memory T cells and memory B cells can be activated quickly to kill off that virus or destroy that virus. Another way a vaccine can work is by containing mRNA. mRNA strands carry the code to make certain proteins, or in other words, viral antigens. So this is how the COVID vaccines work. They are mRNA vaccines. They contain a small strand of mRNA. When that mRNA is injected into your body, it's taken up by your tissue cells. Your tissue cells will naturally read that code on the mRNA and synthesize the viral antigens, or just those viral proteins. Those viral antigens will then be recognized as foreign by your body, and your body will then produce memory B cells and memory T cells to recognize those foreign viral antigens and therefore protect your body if you were ever exposed to the COVID virus that actually contains those viral antigens. So it allows your body to recognize the COVID virus in the future without ever actually having to be exposed to the actual virus. So we've talked a lot about antigens versus antibodies, and let's make sure that we understand the difference between these two terms. Antigens are any molecule, usually a protein, that initiates an immune response. Antigens can be large and complex molecules, or they might be more simple. So here we have a virus. This is a type of pathogen, right, that can infect the body. And here on the surface, you see that it can have many different types of antigens. Here we have a virally infected cell. So this cell was actually infected by a virus, and it's presenting on its surface some of those viral antigens. Now, the antibodies will bind to specific regions on the antigens called epitopes. So epitopes are also known as antigenic determinants. These are the specific regions on an antigen where antibodies bind. For example, here I have a bacterium. On its surface, it has some proteins on it. These are the antigens, right? The foreign proteins. Now, each one of these antigens actually has several different epitopes on it. So each one on this picture has 2 epitopes. So it has 2 specific regions that antibodies can bind. So you can see one epitope here and another epitope here. So this demonstrates that multiple antibodies can bind to a single antigen as long as there are several different epitopes or binding regions on that antigen. So this also means that the more antibodies or the different types of antibodies that your body produces to recognize a specific antigen, the better your immune system will be able to respond. Let me bring up another term related to antigens known as haptens. Haptens are small molecules that only become antigenic when they bind to larger proteins in the host body. For example, the chemical in poison ivy that causes allergic reactions is a hapten. Penicillin is also another example of a hapten. I like to think of haptens as being like half an antigen. Okay, so a hapten doesn't actually become antigenic or cause an immune response until it binds to a larger protein, and therefore then it becomes a full antigen. And let me give you an example of this and how it works with poison ivy. Okay, so here you can see a poison ivy reaction. This is actually my arm. I'm very allergic to poison ivy. And so here you can actually see the blistering that results from exposure to the oils on the poison ivy. So your immune system, when you're exposed to poison ivy, your immune system starts to attack your skin cells. And that's what causes all the blistering. And then here you can see over time, the arm will start to heal. Why does this reaction occur? Well, let's look at what's in poison ivy. So if you come in contact with the roots or the leaves of the poison ivy, what happens is it releases certain oils. Within the oils, there's a specific chemical in there called urushiol, which is a hapten. By itself, urushiol will not cause an allergic reaction or an immune response. Okay, however, when this poison ivy oil comes in contact with your skin and you let it sit there for a while, that's whenever the reaction is going to happen. So here, let me just show you an example of what happens immediately after being exposed to poison ivy. So right away, you can see the urushiol molecule sitting on the skin cells. Okay, at this point, if any T cells come along, or any immune cells come along, they're not going to recognize the urushiol as being anything foreign. So your immune cells are going to say, everything looks good here. However, if you do not wipe that poison ivy oil off immediately, over time, what ends up happening is some of those urushiol molecules will actually bind to the membrane proteins on your skin cells, creating an unfamiliar antigen. And your body will then respond to that unfamiliar antigen now that's on your skin cells by attacking your skin cells. So your immune cells will come along and then all of a sudden recognize that as something foreign, and it's going to say it's a foreign cell attack. And your body actually thinks that your skin cells are foreign cells because the urushiol has binded to the proteins on your skin cells and created something that appears foreign. So again, urushiol is a hapten. By itself, it is not antigenic. It does not cause an immune response. But it's only after urushiol binds to a larger protein in your body that your immune system recognizes that as foreign and then causes an immune response. So this is why, again, if you wipe off the poison ivy oil pretty quickly after exposure, it lessens the chance of your body having an immune response. However, if you let it sit in your skin too long, that can result in the immune response and blistering that you see in this image here. Now let's talk a little bit more about antibodies. Antibodies are also known as immunoglobulins. They are proteins. They're usually Y-shaped or T-shaped. As here, you can see that they are 2 Y-shaped antibodies. And your antibodies are actually made by specific lymphocytes called B cells. Antibodies bind to antigens to help the immune system neutralize or recognize a pathogen. So here we have an antigen, a foreign protein shown in blue. It has 2 epitopes or binding regions to it where you can see 2 different antibodies binding to the different epitopes. Now let's look at the structure of antibodies. Antibodies have two major regions to them. The one region is known as the constant region, and you can see that pictured in red here. This is the region of an antibody that is the same among antibodies of a given class. So there are actually 5 classes of antibodies. We have immunoglobulin A, D, E, G, and M, each with their own chemical properties. All of the antibodies that belong in immunoglobulin A class, they all have the same constant region. Okay, for example, all the antibodies that would be part of immunoglobulin D class, they would all have the same constant region. And that's what gives them their certain chemical properties. However, what differs between the antibodies, for example, that are all within the class immunoglobulin A, what differs between the different antibodies is their variable region. Okay, the variable region, as you see here and here in purple and black in this image, the variable region is a unique region of an antibody containing the site where the antibody binds to the epitope of an antigen. So these two antibodies that I have pictured here have the same constant region, so they're going to be part of the same class. However, they have a different variable region, which means that they would each bind to a different epitope of an antigen. Now let's look at the 3 cell groups involved in adaptive immunity. So these cells are either involved in cellular immunity or humoral immunity. And then we're going to actually look at cellular immunity by itself, as well as humoral immunity by itself, and look more at how these cells are involved in these different types of immune responses. So the 3 cell groups involved in your adaptive immune response include T cells, antigen-presenting cells, or APCs, and B cells. T cells are a type of lymphocyte. They are primarily involved in cellular immunity, but you'll see that helper T cells are involved in both cellular and humoral immunity. So where are T cells produced? T cells are produced in our red bone marrow. And then they actually mature in an organ called our thymus. After they mature, they then are deployed to our lymphatic tissues and organs, where they will then become exposed to specific pathogens and so forth. Next, we have antigen-presenting cells. Antigen-presenting cells include macrophages, B cells, and dendritic cells. Dendritic cells are immune cells found in tissues contacting the external environment, so like our skin as well as our mucous membranes. Antigen-presenting cells present antigens to helper T cells. So that is their job. They're called antigen-presenting cells because they present antigens. And specifically, they're presenting antigens to helper T cells, as you see pictured down here. Antigens are presented on the surface of antigen-presenting cells in a membrane protein complex called MHCs, which stands for major histocompatibility complexes. So here you can see an antigen-presenting cell. It could be a macrophage, a B cell, or a dendritic cell. On its surface in blue, you see an MHC2 protein. Okay, so this is the major histocompatibility complex. And bound to that complex is a foreign antigen. And this antigen is being presented on the surface of the antigen-presenting cell so that helper T cells can come along and recognize those foreign antigens. If that helper T cell recognizes that antigen as foreign, it's then going to initiate or stimulate an immune response. And we'll look at how that happens later on in this lecture. Now, the last group of cells involved in adaptive immunity are the B cells. B cells are a type of lymphocyte. They are primarily involved in the humoral immunity. And they are produced in the red bone marrow, and they actually mature in the red bone marrow. Once they mature, they are deployed to similar locations as the T cells. They are deployed to our lymphatic tissues as well as our lymphatic organs. So here in this image, you can see how antigen-presenting cells are able to present antigens on these MHC complexes. So here we see an antigen-presenting cell. This is a macrophage. Here we see some bacterium. The bacterium is being engulfed by the macrophage through a process known as phagocytosis. Once the macrophage pretty much eats the bacteria, it's going to, that vesicle that it uses to like bring the bacteria into the cell is then going to fuse with lysosomes. Lysosomes are specific organelles within a cell that contain digestive enzymes. So whenever the lysosome fuses with the vacuole containing the bacteria, it releases digestive enzymes that allow the macrophage to pretty much break down and digest the bacteria. So some of the proteins from that bacteria will then bind to these MHCs and then move to the surface of the cell. And this will therefore allow the macrophage to then present some of those antigens from the bacteria on its surface. Okay, so this allows the body to monitor what type of pathogens or what type of proteins are present in the body. Now let's test your understanding of the topics that we covered so far. So pause the video and try to answer these questions by writing down the term that is being described. All right, number 1, the name given to cells such as macrophages, B cells, and dendritic cells that phagocytize pathogens and cells and then present antigens on their surface. That would be antigen-presenting cells, or APCs. Number 2, the region of an antibody that is similar among all antibodies of a given class. That would be the constant region because it's constant among all the antibodies of a given class. Number 3, the region of an antibody that contains the antigen binding sites. That would be the variable region. Okay, the variable region is called variable because it differs between different antibodies, and each antibody will be able to, or each type of antibody will be able to bind to a specific type of epitope or specific region of an antigen. Number 4, these are a group of lymphocytes that are primarily involved in cellular immunity. That would be the T cells or T lymphocytes. Number 5, these are the sites on an antigen where antibodies bind. That would be the epitopes. Number 6, this term is used to describe molecules that are not antigenic until they bind to larger molecules in our bodies. That would be haptens. I remember this again by thinking that haptens are half of an antigen. Number 7, this group of cells is primarily involved in humoral immunity. That would be the B cells, or also known as B lymphocytes. Now let's look at the type of T cells that are involved in cellular immunity and look at their different roles in cellular immunity. We have cytotoxic T cells, helper T cells, regulatory T cells, and memory T cells. Cytotoxic T cells are also known as killer T cells, and that is because they are the effector cells of our cellular immunity. They are the actual cells going out and killing other cells. Cytotoxic T cells, they respond to foreign antigens that are presented on MHC1 proteins. MHC1 complexes are actually found on all nucleated cells of the body, so that's an important thing to remember. MHC1 complexes are found on all the nucleated cells in your body. So every cell in your body that has a nucleus has MHC1 complexes on it. Whereas MHC2 proteins are only going to be found on antigen-presenting cells. Cytotoxic T cells, again, they're called killer T cells as well because they directly kill foreign and diseased cells by using two types of chemicals. Perforins, which perforate the cell membrane of the diseased or foreign cell. And then they also use granzymes, which pretty much, digest the foreign cell or diseased cell. These two chemicals are very similar to the chemicals that are used by natural killer cells, which you saw are involved in our innate immune response. The second major group of T cells involved in cellular immunity are called our helper T cells. Helper T cells, help out the adaptive immune system. Okay, that's why they're called helper T cells. What do they specifically do? They respond to foreign antigens presented on MHC2 proteins. These MHC2 proteins are only going to be found on antigen-presenting cells like B cells, macrophages, and dendritic cells. Helper T cells function by secreting chemicals called interleukins. Okay, interleukins, inter means between, leukins means between the leukocytes or white blood cells. So these chemicals help communicate between different white blood cells. And what these chemicals specifically do is they help to stimulate our T cells, our B cells, and our macrophages, and therefore increase our immune response. The next group of cells involved in our cellular immunity are regulatory T cells. These are also known as suppressor T cells. They help to suppress or inhibit other immune cells like B cells and T cells. So they help to slow down our immune response. Regulatory T cells are going to be very important in preventing chronic inflammation as well as autoimmune diseases. And then finally, we have our memory T cells. Memory T cells are long-lived T cells. They can survive in your body for decades. And their specific job is to help your body remember a specific antigen so that your body can have a quick immune response if you are exposed to that pathogen again. So if you ever re-encounter a pathogen, these memory T cells will quickly initiate an immune response, even before you actually get sick. Okay, so let's summarize some key points about the MHC proteins or MHC complexes. All right, so we actually had MHC1 proteins and MHC2 proteins, and each one has a different job and are found in different places. Okay, so cytotoxic T cells, what type of cell is going to trigger their response? Well, all nucleated cells in our body that are either infected, also any type of foreign cell. Cytotoxic T cells, they respond to MHC1 proteins. Helper T cells are only going to respond to antigen-presenting cells, and these antigen-presenting cells have what we call MHC2 complexes on their surface. So here you can see some cytotoxic T cells killing a cancer cell. And then here you actually see a helper T cell binding to the surface of a macrophage. Macrophages are antigen-presenting cells. And so here this helper T cell is investigating some of the MHC2 proteins on the surface of this macrophage. So there's 3 different things that pretty much happen after our body is exposed to a pathogen. These stages are going to be very similar in cellular and humoral immunity. So let me list out the 3 R's. The first thing our immune system is going to have to do is recognize the pathogen. Okay, and our body's going to do this by recognizing the foreign antigens within our body, so the foreign antigens on those pathogens. Next, our immune system is going to react. The immune system will destroy the pathogens, foreign cells, or disease cells. And then finally, our adaptive immune system will remember the pathogen. So our adaptive immune system will form memory T cells or memory B cells. These will persist in the body, so the body can then remember the specific antigens that were encountered. The next time you're exposed to that pathogen, your body will be able to initiate an immune response much quicker and therefore actually usually prevent you from even getting sick. So let's look at how these 3 stages are involved when we looked at cellular immunity. Okay, so the first thing that has to happen is our body must be able to recognize the pathogen or the antigens, right? So there's 2 ways that our cells that are involved in cellular immunity will recognize the antigens of a pathogen. First off, the T cells will identify foreign antigens presented on the MHC proteins. And we actually have 2 different types of MHC proteins. So there's 2 different ways, again, that we can recognize these antigens. So any antigen MHC1 complexes found on all nucleated cells will activate our cytotoxic T cells. So here you can see an infected cell. This cell is infected by a virus. It's presenting some of the viral antigens on its MHC1 complex. So we can call this an antigen MHC1 complex, right? Because the antigen is bound to the MHC1. What's going to recognize that? The cytotoxic T cells. Here we have an antigen presenting cell that has phagocytized a viral virus, and it's presenting some of the viral antigens in its MHC2 proteins. So we could call this an antigen MHC2 complex. It's going to be found on the antigen presenting cells. And again, that's going to activate helper T cells. Okay, so let's look at a little bit more at the MHC1 and MHC2s to understand kind of how our body can recognize foreign pathogens. So here we actually have a normal skin cell. and it's presenting a self-antigen on its surface in the MHC1 complex. Here we have a skin cell that has become cancerous. So some of the proteins that it's making are going to be somewhat abnormal, okay, because it's in cancerous cells, the genes have been mutated. Okay, so here we can see some foreign or abnormal antigens on the surface. The cytotoxic T cells will investigate these antigen MHC1 complexes. If there's a self-antigen present, the cytotoxic T cell will ignore that cell. However, if there is a foreign antigen present in that antigen MHC1 complex, the cytotoxic T cell will then kill the cell and recognize it as being foreign. Now look over here, let's look at how helper T cells can recognize these foreign antigens. So macrophages, their job is to phagocytize dead cells as well as foreign cells and things like that. So if your macrophages are destroying like a dead skin cell, you wouldn't want your body to initiate an immune response in that case because, there's nothing foreign or dangerous happening in the body. So here you can see this macrophage is breaking down this dead skin cell. It's destroying it, and then some of the proteins from that dead skin cell are being presented on its surface, and it's MHC2 proteins. If a helper T cell comes along and investigates this antigen MHC2 complex, it should ignore it. There should be no response because your helper T cell should be able to recognize that as being, a self-antigen, something that is familiar to your body. However, this macrophage on the right here has ingested a bacterial cell, and it's broken that down, so now it has a foreign antigen in its surface. If a helper T cell comes over and recognizes that this antigen MHC2 complex contains a foreign antigen, it will then release chemicals. Remember, we call them interleukins. It's going to release those chemicals and therefore stimulate cytotoxic T cells, B cells, and macrophages in our body, therefore initiating a strong immune response. Okay, so the recognition phase is first recognizing the foreign viral antigens, and then the stage that occurs after that will be a reaction. Okay, helper T cells will stimulate other immune cells such as cytotoxic T cells, B cells, and macrophages to take action. Okay, so here you can see stage 2. The helper T cell has recognized a foreign viral antigen. It's going to stimulate other immune cells like B cells, macrophages, as well as cytotoxic T cells. One way that it can stimulate the cytotoxic T cells is to cause them to proliferate and divide and multiply. And therefore, we have a lot of cytotoxic T cells that can therefore go around the body and kill any of those pathogenic cells or infected cells within the body. You can also see that the helper T cells are stimulating the macrophages to help break down or destroy, phagocytize any of the infected cells. And then the B cells will also be activated. So helper T cells not only stimulate our cellular immunity, they're also going to stimulate our humoral immune response as well. So here you can see the cytotoxic T cells are destroying any of the infected cells. They're doing that by releasing 2 types of chemicals called perforins and granzymes. And then finally, once the infected cells have been destroyed and the amount of viral pathogens in the body has decreased, regular T cells will then release chemicals to kind of suppress the immune response and slow it down. Then after that infection has been cleared from the body will then have what we call memory T cells, or these long-lived memory T cells. They're going to remain in the lymphatic system and therefore stay in your body to help your body better respond to that specific virus if you're exposed to it again in the future. Now let's look at the cells involved in your humoral immunity. So let's first look at the B cells. B cells produce antigen-specific antibodies. Note that their antibodies stay adhered to their membrane. So here we have a B cell. It's producing antigen-specific antibodies on its surface. So each type of B cell will produce a specific type of antibody. B cells also act as antigen-presenting cells by displaying foreign antigens on their MHC2 proteins. So they also can phagocytize pathogens and then present those foreign proteins on their surface, just like macrophages do. Some B cells will become specialized cells called plasma cells, which I'll talk about in a second over here. But these plasma cells are really important because they actually will secrete the antibodies into our blood plasma and our lymph so that they can circulate throughout our body. And I always remember that plasma cells secrete the antibodies into our blood plasma and our lymph because, blood plasma and plasma cells both have the word plasma in their name. Now, helper T cells are also involved in humoral immunity, just like they were involved in cellular immunity. Helper T cells will bind to antigen MHC2 complexes on the B cells, and that will then stimulate the B cells to start becoming plasma cells. Now, plasma cells are going to be really important in humoral immunity because they develop from the B cells and they secrete large amounts of antigen-specific antibodies into our blood plasma and our lymph. Just to give you an idea of how many antibodies these plasma cells are secreting into your bodily fluids, it's estimated that each plasma cell will secrete about 2,000 antibodies per second. That's crazy when you think about that. Okay, this is what allows your antibody levels to increase drastically so that your body, your immune system can better recognize the pathogens and tag them for destruction. After your body has destroyed a pathogen, then what's going to be left in your body are these memory B cells. These are long-lived B cells that can, live in your body for decades. And then the benefit of having these memory B cells is that they can quickly develop into plasma cells if an antigen is re-encountered in the future. So let's look at how the 3 stages of immunity are involved when we're looking at humoral immunity. So let's look at how we can apply those 3 R's that we talked about before. So here we're going to look at an example where the body's infected by a viral pathogen. Here in stage 1, we can see a B cell has phagocytized a virus, broken down that virus, is presenting some of the viral antigens on its surface in this MHC2 protein. You can also see that this B cell is producing some antibodies to help recognize antigens on that specific virus as well. So the stage 1 would be recognizing the pathogen. The B cells will act as antigen-presenting cells by engulfing antigens and presenting them on MHC2 proteins. Helper T cells will bind to the antigen MHC2 complexes on the B cells, as well as other antigen-presenting cells within the body, like macrophages. The next stage will be a reaction. The helper T cells will stimulate the B cells to become plasma cells, which will then secrete antibodies into your bodily fluids, such as your blood plasma and your lymph. These antibodies will bind to antigens, and you can see that happening here. The antibodies are binding to the antigens on the surface of these viruses. And these antibodies will actually use 4 methods to help the immune system destroy the antigens. These 4 mechanisms include neutralization of the pathogen, 2, what we call complement protein fixation and activation. So it pretty much activates the complement proteins within your innate immune system. It will also promote what we call agglutination and precipitation, which will help with phagocytosis. And I'll talk about that in the next slide. After the antibodies have helped the immune system recognize and respond to the pathogen, what's going to be left in the body is our last stage, which is remembering the pathogen. So what's left in our body are these long-lived memory B cells. These B cells will remain in the lymphatic system and therefore stay in our body to help us increase our production of antibodies quickly if we are exposed to that virus again in the future or pathogen. As I mentioned, there are 4 methods used by antibodies to inhibit antigens. Okay, so how do antibodies actually get rid of the antigens or help our body get rid of them? Here's the 4 methods. The first is neutralization. So what happens is our antibodies will bind to the antigens on a pathogen and pretty much block it from infecting the body. A great example of neutralization would be the rabies post-exposure shots. When you've been exposed to rabies, you'll get these post-exposure shots that contain specific antibodies. Those antibodies will bind to the antigens on the rabies virus and prevent the rabies virus from being able to infect your cells, and therefore it can neutralize the virus. Another thing that antibodies do is they help to activate complement proteins. So here you can see an antibody. It's fixated on a complement protein, meaning it's bound to it, and it's activating it. Activated complement proteins, I talk about in the lecture where I talk about the innate immune system. Well, they actually help to destroy pathogens by promoting 4 things. Complement proteins help to promote inflammation. They help to promote immune clearance, so help your body get rid of the antibody antigen complexes that are formed. It helps promote phagocytosis and also helps to promote cytolysis, or the destruction of diseased or pathogenic cells. And then finally, antibodies help with what we call agglutination and precipitation. Agglutination and precipitation are very similar in that what we're doing is we're causing things to clump up. With agglutination, what we're doing is we're clumping up cells. So this could be bacterial cells or foreign red blood cells. So here you can see an example of agglutination, where the cells are being clumped up together because the antibodies are linking the cells up together. This is going to make it easier for phagocytosis to occur, right? So this macrophage could come over and engulf all of these pathogenic cells or foreign cells very easily if they're all clumped together. Okay, so agglutination is the clumping up of cells. Precipitation is the clumping of small particles like viruses or toxins. So here we can see some viral particles that are clumping up together because the antibodies are linking them together. And that's going to also help promote phagocytosis and make it much easier for macrophages and other phagocytic cells to come along and actually just eat up all those viruses or toxins in one giant clump versus if they were all scattered throughout the body. Okay, so hopefully now you understand kind of the steps involved in cellular immunity and humoral immunity. One thing I want to talk about is the difference between what happens in our body when we are first exposed to an antigen versus the second time we're exposed to an antigen. The first time you're exposed to an antigen, we call that your primary immune response. The second time you're exposed to an antigen, we call that your secondary immune response. So the first time you're actually exposed to an antigen, it takes several days, almost up to a week, before your body starts producing antibodies that can help your immune system recognize that antigen. And it also, it takes a while, and the level of antibodies is not that great. Okay, so you can see the level of antibodies increases, but then it will decrease over time. However, when you're exposed to that antigen a second time, your body already has memory B cells present. So the second time you're exposed to that antigen, your body can quickly, remember those memory B cells can become plasma cells, and then they'll start secreting a whole bunch of antibodies right away. So your body will respond very quickly by producing a large amount of antibodies. Now, there's a lot of different types of antibodies. Here you can see that there's 2 different classes of antibodies being produced when you're exposed to an antigen. The immunoglobulin G antibodies are pretty much what we can call long-term antibodies. They stay in your body for a very long time. So after the second time being exposed to an antigen, your body will make a whole bunch of these long-term antibodies, and that creates long-term immunity. So now you have this high concentration of antibodies that's circulating in your body for a long time, providing you long-term immunity against a certain virus, for example. This is why after you get like your first vaccine, you usually get a booster, right? So after we got our COVID vaccines, the first one, a couple of weeks later, you had to get a second one. And that is to help promote your secondary immune response. And sometimes it might even require a third shot to help further promote production of those antibodies. And what your goal is to produce a whole bunch of these long-lasting antibodies to give your body the best chance of being able to recognize that pathogen if you're exposed to it, so your body can fight it off quickly before you even get sick. All right, so again, we have 2 different types of responses depending on whether it's your first encounter with a pathogen or antigen versus whether it's your second. The primary response is the reaction of the adaptive immune system after the first time it is exposed to an antigen. The primary response is usually very slow and weak because it takes time for your naive B and T cells to be activated and to proliferate. So it takes a while for your body to start creating those memory T cells and B cells, and for your B cells and T cells pretty much to learn to recognize those foreign antigens. During your primary immune response, there are few antibodies that are made, and the antibody levels tend to decline shortly after infection. However, one of the benefits of the primary immune response is what you're left with are memory B and T cells. This helps your body respond during your secondary immune response. This is the reaction of the adaptive immune system after exposure to an antigen the second and subsequent times. The secondary response is quick and strong because you have these memory B cells, for example, that can immediately be activated. They'll become plasma cells, which will then start to pump out a whole bunch of antibodies. The large amount of antibodies are produced and the levels remain high for a long time, giving you long-term immunity. The pathogen can then be destroyed before you even get sick. Your body can respond so quickly when you're exposed again to these pathogens because those antibodies help your body quickly recognize and then react to the antigens that are present. The last thing we're going to look at in this lecture video are immune system disorders. Hypersensitivities are an immune response that is too vigorous. Hyper means higher, so your body is too sensitive to a certain antigen. Examples of hypersensitivities include allergies. Allergies are actually an inflammatory response to chemicals in the environment or food that are exaggerated. If those antigens or chemicals come in contact with your blood, that can lead to anaphylactic shock. This is a quick and severe allergic reaction, usually caused by allergens entering the bloodstream. An example of this could be exposure to like peanuts or something like that. So once you start ingesting the peanuts, those chemicals within the food for some people can lead to anaphylactic shock. The symptoms of this can lead to bronchoconstriction, so your airways start to close up, and vasodilation, your blood vessels start to dilate. That causes lowering of your blood pressure. The treatment, if somebody's in an anaphylactic shock, is using an EpiPen. An EpiPen actually contains epinephrine, and if you remember, epinephrine is really important in stimulating our sympathetic nervous system. Okay, so when you inject that epinephrine into the body, it actually stimulates bronchodilation, so it opens up the airways, and the sympathetic nervous system actually stimulates vasoconstriction, so it causes the blood vessels to constrict throughout the body. That helps to increase the blood pressure. Next, we're looking at autoimmune diseases. These are conditions where the immune system attacks the body's own tissue cells because it mistakes the self-antigens on those tissue cells as foreign antigens. So it's called autoimmune because auto means self, right? So your immune system is attacking yourself. Examples of this include lupus. When somebody has lupus, multiple tissues in their body are usually affected. Some inflammatory bowel diseases are the result of autoimmune diseases such as Crohn's disease and colitis. Type 1 diabetes, the body's immune system is attacking the beta cells within the pancreas. We have rheumatoid arthritis and MS, multiple sclerosis, are just some examples of autoimmune diseases. So what actually causes the body to start attacking your own tissue cells? There's actually three main things that usually lead to autoimmune diseases developing. The first one is known as cross-reactivity. This is when your body is exposed to certain viral or bacterial antigens, and this actually causes the immune system to attack self-antigens that are similar in structure. Okay, so let's say you're exposed to a specific bacterial antigen. Your immune system learns to attack that bacterial antigen, but it might become a little bit too generalized, and it actually might start attacking like self-antigens within your body that are structurally similar to those bacterial antigens. And that can lead to your body attacking your own tissue cells. Another thing that can lead to autoimmune diseases are abnormal exposure to self-antigens to the blood. Some self-antigens normally do not contact the blood, and when they do, it triggers an immune response. An example of this would be the production of anti-sperm antibodies leading to sterility. The sperm cells in males should never come in contact with the blood within males. Okay, they're actually protected by what we call the blood-testes barrier. However, trauma to the testicles or something like that could cause the sperm cells to come in contact with a person's blood. That would then trigger the immune system to start producing antibodies or T cells that can then attack those sperm cells. And then what ends up happening is that the immune system then starts killing the sperm cells, leading to a lower sperm count. And that can potentially lead to sterility. And then finally, we have mutation of self-antigens as a possible cause of autoimmune diseases. Viral and bacterial infections may actually change the structure of our self-antigens, making them appear foreign. Many times when you're infected by viruses or bacteria, that can actually cause mutations in your genes. And those mutations in your genes cause your body to make foreign proteins. And therefore, your immune system will then start attacking your cells that are producing these foreign proteins. But again, those mutations were caused initially by some type of viral infection. And then finally, let's look at what we call immunodeficiency diseases. These are conditions where the immune system fails to have a strong enough response to a pathogen, and therefore the individual cannot rid the body of those pathogens. Some causes of the development of immunodeficiency diseases include genetics, so you might be born with some type of genetic defect, environmental agents, as well as immunosuppressing drugs. Some environmental agents that can cause immunodeficiency diseases include certain viruses. So let's look at two specific examples that I want you to know. The first is known as SCID, which stands for Severe Combined Immunodeficiency Disorder. This is actually a genetic disorder, and it's caused by the absence of functional B cells and T cells. So this is actually caused by a genetic mutation that therefore causes the person to not have functional B cells and T cells. Well, we know B cells and T cells are both important in our adaptive immune response, right? T cells are necessary for cellular immunity. B cells are necessary for humoral immunity. So if your body does not produce functional B cells and T cells, that means you have no adaptive immune response. So therefore, if you're exposed to a virus or bacteria, your body cannot learn to better be able to fight off that pathogen in the future when you're exposed to it again. Also, because you only have the innate immune system, you don't have functional cytotoxic T cells, you don't have functional B cells that can make antibodies, so you pretty much have a depressed immune system. So it makes it harder for your body to fight off those pathogens. And therefore, somebody who is born with this disorder can easily die from some common infections that other people would be able to fight off. Another example of an immunodeficiency disease is AIDS, which stands for Acquired Immunodeficiency Disorder. Now, this is acquired whenever you're exposed to the HIV virus. The HIV virus specifically infects helper T cells. Here you can actually see the HIV virus particles emerging from an infected helper T cell. So what happens then when the HIV virus infects these helper T cells is it slowly destroys them. We know that the helper T cells are extremely important in stimulating our immune system, right? So if you do not have functional helper T cells, well, then you have a suppressed innate immune response because the macrophages aren't being stimulated. You have a suppressed cellular immune response because the T cells are not being stimulated. And then you have a suppressed humoral immune response because B cells are not being stimulated to become plasma cells. So again, pretty much your immune system is not functioning properly at all if you do not have these functional helper T cells. All right, so let's end this PowerPoint by quickly reviewing the characteristics of the innate and adaptive immune system. Tell me whether each one of these characteristics falls under innate immunity or adaptive immunity. It involves T cells, B cells, and antibodies that are specialized to fight specific pathogens. That would be adaptive immunity. Provides nonspecific resistance against a broad range of pathogens. That would be innate immunity. Includes our first line of defense. This would include our skin as well as our mucous membranes. That would be our innate immunity. includes our second line of defense. That includes a lot of different things, such as some of our white blood cells, as well as fever, inflammation. Also, we have some proteins that we call antiviral proteins known as interferons, as well as complement proteins. They're all part of our second line of defense. That would be our innate immunity. Okay, they fight a broad range of pathogens, and they do not adapt to become better at fighting a pathogen the second time your body's exposed. This includes our third line of defense. That would be our adaptive immunity. Our third lines of defense include our cellular immune response and humoral immune response. This includes fever and inflammation. That would be innate immunity. Those are more generalized responses to pathogens. This can create a memory of a pathogen. That would be our adaptive immunity. This becomes more efficient at fighting a pathogen the more it is exposed to that pathogen. That's our adaptive immune system. And then this has more of a localized effect on the body. That would be our innate immune response. And its effects on the body are usually widespread or systemic. That would be our adaptive immune response. And that's due to the fact that T cells and B cells are found all throughout our system, especially in our lymphatic organs. And then antibodies circulate through our blood plasma and our lymph, therefore providing widespread protection. In the remainder of this lecture video, I have some practice questions or review questions that you can do to help test your understanding of the topics that we cover. What I'm going to do is on each of the activity slides, I'll tell you to pause the video, and then I will put up the answers so that you can check your answers. So you can go through each one and do this on your own to see if you can answer the questions. So in this first one, you want to list the 4 ways of acquiring adaptive immunity and give examples of each type. Here is the answer key for that question. Next, let's review the stages of cellular immunity. Try to fill in all the blanks below. Here is the answer key. Next, you're going to be reviewing the stages of humoral immunity. Try to fill in the blanks below. Here is the answer key for that. And then finally, see if you can fill in the blanks below to help you test your understanding of the immune disorders that we covered. Here is the answer key. All right, that is the end of the lecture on adaptive immunity. Please send me a message if you have any questions on the content that I covered in this lecture video.